Hello students, today we will study about the heterospory. The production of one kind of spores that germinates to produce bisexual gametophytes is called the homospory. The early land plants, bryophytes except few, are homosporous. As opposite to this, the production of two different kinds of spores, two different kinds of spores. The smaller one is the smaller microspores producing male gametophytes, and the larger second is the larger megaspores producing female gametophytes is called heterospory. The structural and functional heterospory has been reported in 9 genera. कौन कौन से? Selaginella, Isoids, Stylites, Marsilia, Pleureria, Reginiladium, Salvinia, Azolla, Platygoma of the Pteridophytes, of which the Heterospory, and out of these 9, in this we study or discussed the Heterospory of Selaginella. That is the life cycle of the Selaginella. That is the microspores, megaspores, Heterospory in Selaginella. All the species of Selaginella are heterosporous, means they produce two different kinds of spores in each plant. In each plant, these spores differ with each other mainly in respect of their size and function. The smaller one, named the microspores, are produced in large numbers in microsporangia, and the larger ones, named the megaspores, are produced in smaller numbers inside the megasporangia. The initial development of each kind of spores inside the sporangia is the same, दोनों का सेम है, but the difference starts with the formation of spore mother cells. जैसे ही spore mother cells का बनना स्टार्ट होता है तो उनमें डिफरेंसेस होना भी स्टार्ट हो जाता है. In the microsporangia, all the microspore mother cells undergo meiosis and form the microspores. Therefore, they are produced in large numbers. Therefore, they are produced in large numbers and their size also remains small. On the other hand, all the spore mother cells in megasporangium do not complete their development, means only one or a very few megaspore mother cells undergo meiosis and produce a very small number of functional megaspores, usual 4. Consequently, the megaspores become much larger in size and store more reserve food as compared to microspores. The microspores and megaspores not only differ in size but also differ in their function. Each microspore germinates in situ and produces a male gametophyte. The vegetative part of male gametophyte is represented only by a single prothelial cell. Single prothelial cell. The remaining portion of male gametophyte becomes an antheridium, which produces 8 art sterile jacket cells and 128 to 256 anthrozoids. Anthrozoids. The megaspore also germinate in situ and produces a female gametophyte. The female gametophyte has a large number of vegetative cells filled with reserve food materials so that they can provide a proper nourishment to the developing embryo. Next, the degeneration of non-viable megaspore mother cells and the formation of functional megaspores has been worked out by Petit 1971. According to him, the viable mother cells are larger, whereas non-viable mother cells are smaller in size. In addition, the viable ones are characterized by the aggregation of mitochondria, plastids at the prophase and contain intranuclear vesicle in their nuclei, and the non-viable ones, on the other hand, possess lysosomes. Similarly, the viable megaspores inherit the mitochondrial plastids from the mother cells, whereas the non-viable megaspores mother cell do not. However, Brooks in 1973 indicated that the formation of megasporangium in Selaginella is under the control of ethylene. Ethylene is a hormone that retards cell division. Cell division. That retards cell division. Next, that is the origin of heterospory. The origin of heterospory can be discussed on the basis of evidences, and these evidences are the three evidences. First is the paleobotanical evidences. Second is the developmental studies. Third is the environmental studies. First, paleobotanical evidences or studies. It has been suggested that heterospory arose due to degeneration of some species in a few sporangia. Due to degeneration of some spores, not species, due to degeneration of some spores in the sporangia. As more nutrition become available to less number of spores, कि उनका degeneration क्यों होता है spores का, क्योंकि जो बचे हुए स्पोर्स है उनको न्यूट्रिशन अधिक क्वांटिटी में गेन हो सके। Means the surviving spores grow better, hence increase in the size. Paleobotanical evidences show that the earlier vascular plants were all homosporous and the heterosporous conditions appeared subsequently in the lowermost upper Devonian. A number of heterosporous genera belonging to the Lycopsida, Spinopsida, and Pteropsida were known in the late Devonian and early Carboniferous periods. During this period, important heterosporous genera कौन से लेपिडोकार्पोन, लेपिडोस्ट्रोबस, मजोकार्पोन etc. Calemocarpon, Calemostix, Paleostix, these are. Some of these forms even arrived at the seed stage. According to Williamson and Scott in 1894, two species of Calemostix form the initial stage that might lead to the Heterospory. In Calemostix or species and the means in Calemostix की species Calemostix biniana, most of the sporangia were with large number of small spores in tetrads, but in some sporangia spores were large. Next evidence, that is the developmental studies. In heterosporous pteridophytes, the development of micro and megasporangia follows the same pattern. They have identical organization but for their size. While in, megasporangia, most of the spore mother cells degenerate, but in microsporangia, only a few spore mother cells or microspore mother cells may disorganize, only few. The phenomenon of heterospory becomes distinct either before or after meiosis. In the microsporangium, all the sporocytes undergo meiosis and form a large number of the microspores. However, in megasporangium, a part of the sporogenous tissues degenerate and they provide nutrition to growing megaspores. In isoids, there are only 50 to 300 megaspores in megasporangium. Next, in Marsilia, Salvinia, and Azolla, the phenomenon of heterospory becomes distinct after meiosis. In Marsilia plant, 64 microspores and 64 megaspores are formed after meiosis in microsporangium and megasporangium, respectively. In microsporangium, all the microspores are functional, while in megasporangium, one megaspore is functional and the rest degenerate. Next study, that is the experimental. Experimental studies on Selaginella by the Goebel, Goebel 1905, and Marsilia by the Stuck Shattuck in 1910 suggest that nutritional factors mainly govern the heterospory कि जो न्यूट्रिशनल फैक्टर्स हैं वो हेट्रोस्पोरी को गवर्न करती है, कंट्रोल करती है. अंडर कंडीशंस ऑफ लो लाइट इंटेंसिटी द फोटोसिंथेटिक activity of Selaginella was retarded and it produced microsporangia. While in Marsilia, sudden lowering of temperature, the size of microspores in sporocarp increases by 6 times. Next, that is the biological significance. The heterospory is of great biological significance on the following facts. कौन कौन से? First, that is the development, or one is the development of the female gametophyte starts while the megaspore is still inside the megasporangium. Still inside the megasporangium. Second, same is true of microspores, that is, they start germinating into male gametophytes while they are still inside microsporangium. Next, the female gametophyte derives its nourishment from the sporophyte, that is, the female gametophyte is dependent on sporophyte for its nourishment. Next or last, the dependence of female gametophyte on sporophyte for its nourishment provides better starting point for the development of new embryo than an independent green prothallus which has to manufacture its food itself. Next, that is the seed habit. The adaptation of heterospory and the retention and germination of single megaspore within the megasporangium. Retention of single megaspore within megasporangium to form a female gametophyte led to the phenomenon of seed habit. That is a characteristic feature of the spermatophytes. A seed is that ovule which contains an embryo developed as a result of fertilization. The origin of seed habit is associated on the basis of the following. First, that is the production of two types of spores, means the heterospory. Second is the reduction in the number of megaspores. Finally, reduction of megaspores, number of megaspores finally to 1 per megasporangium. Next, retention and germination of the megaspores and fertilization of the egg. Next, continued development of the fertilized egg with the embryo while still in situ. Students, the heterospory is finished in this video, and in the next we will study about the apogamy and apospory. Students, these are the general topics and maybe come up to the heterospory maybe come up to the 5, 6 or 7 marks, and you can give remember the diagram of the life cycle of the Selaginella and put as an example of the heterospory. Thank you.